For our virus presentation, we chose Influenza by Caleb, Sanjana, Meriden, and Jamie.
The life cycle of the flu looks similar to those of other viruses. It starts with entry into the host cell. In the case of the flu, VRNPs, or viral proteins of RNA, enter the cell. The next step is transcription and replication of the viral genome. Next, the viral RNA is exported from the nucleus into the cytoplasm. Assembly of viral components and budding of the host cell plasma membrane happens, where the virus then can spread to other cells. In evolutionary perspective, viruses do not have a good error-catching system during replication like eukaryotic RNA polymerases do. This leads to lots of mutations and hence an always changing and evolving flu virus. The genome of the flu virus contains single-stranded RNA, unlike our double-stranded DNA. It has 8 genes which code for 11 different proteins. Exchange of genetic material for the flu is also similar to a few other viruses. It first attaches to the host cell by binding to the receptors and then enters through receptor-mediated endocytosis, so it is in an endosome. It enters the cytoplasm, but ultimately needs to end up in the nucleus. This is possible because the VRNPs discussed earlier have nuclear localization signals which allow it to enter the nucleus. Influenza, however, has something called negative RNA, which cannot be used by cellular machinery to make proteins. For this reason, once it is in the nucleus of the host cell, a viral RNA polymerase transcribes it into positive RNA. This then can be transcribed by the cellular RNA polymerases. After that, it goes back into the cytoplasm where cellular enzymes and ribosomes translate it into proteins. At this point, it has taken over the cellular machinery of the host cell. After the viral components are assembled into functional viruses, they eventually are released from the host cell due to lysis. They are now free to infect other cells.
Mode of infection. Influenza can spread from person to person through direct contact, like shaking hands, hugging, and physical contact. Indirect contact, like touching infected objects, like a door handle or desk, or consuming contaminated food. Or aerial transmission, such as coughing, sneezing, and talking. The most common method of transmission is through exposure to respiratory droplets through sneezing or coughing. Subjects must be in a close vicinity as droplets don't remain suspended in the air. Measures to prevent spreading are to cover coughs and sneezes, wash hands, and not touching hands to the face. New vaccines are made each year for seasonal influenza. Effects of influenza are fever, sore throat, cough, headaches, and fatigue. Nausea, diarrhea, and encephalitis are also common effects to children.
In 2017, an article was published by Laura Beal that discussed a new technique for developing influenza vaccines. The goal of this technique was to develop a vaccine that could protect people from most strains of the flu instead of a certain strain that appears each season. By developing this universal vaccine, the intent was to reduce the frequency that the flu shot was needed so people wouldn't have to get a new one every single year. The reason a flu shot is needed every flu season is because each strain has a different combination of 2 proteins on its surface, hemagglutinin and neuraminidase. The different combinations of these proteins is actually how each strain, like H1N1 or H3N2, are named. Influenza strains are divided into 2 types, type A or B. Type A is usually more difficult to vaccinate against because it's constantly mutating and even swaps genes with other strains, which is how they can sometimes jump from animals to humans. In order to protect against all of these mutating strains, researchers are working towards developing a vaccine that would create antibodies against the stem of the protein, which stays relatively the same no matter what combination of proteins surrounds the cell, instead of antibodies that protect against a specific combination of the heads of these proteins.
So for how this influenza virus and presentation are relevant to our current coursework in this class, we briefly in class talked about viruses and this chart seen on this slide that show how viruses will infect host cells, amplify, and then leave and spread to other cells, and that is how it is spread. And this chart is from the textbook, and it shows visually how that happens within host cells. And we also learned in class about both transcription and translation, and they are both processes used by the flu virus to infect and then take over and spread in other host cells. And also, it's not relevant to our learning as much as our process of learning, but currently we've had a lot of changes in our coursework in every class in the whole university due to the current viral pandemic, and this virus acts very similarly to the flu virus in that it has similar symptoms and it spreads similarly. So hopefully in the future we will be able to not have something like this affect our coursework like this, because we can prevent it better and keep everyone safe and keep these viruses away so we can learn about them, such as we just did this influenza virus.